Chapter 12, Muscles. In this chapter, we're going to focus on skeletal muscle, look at body movement, and then we will end with smooth muscle. So to start off, let's make sure we understand our three types of muscles. So we have our skeletal muscle. These are the muscles that you find in your biceps, your triceps, etc. These are unique because they are voluntary. So you can actually control your skeletal muscle contractions. They are also striated. If you see, you have these little stripes, and they are multinucleated. So most cells, or all cells that have a nucleus, only have one, and it's in the center of the cell. Skeletal muscle fibers have multiple nuclei, and they're not in the center, they're out on the edges near the sarcolemma. What else? They're contractile as well as all other muscles being contractile, but that makes it different than other types of cells. Another thing is that they're very long. So these are very long cells. The other type of muscle are cardiac muscles. So cardiac muscles, if you take a look, are also striated. So these striations occur because these muscles have sarcomeres that are highly organized. They have a central nuclei and they just have one, but these muscles are found in your heart, and there are these structures called intercalated discs. And these are important because you have gap junctions here. And gap junctions allow the transfer of electrical impulses from one muscle cell to another. And then smooth muscle is found in all of your visceral organs. So you'll find it, for example, in your blood vessels, in your respiratory tract, in your GI tract, your reproductive tract. So basically everywhere else. And they're called smooth because when you look at it, they don't have those striations. So they look smooth. And they don't have those striations because they don't have sarcomeres, which means their actin and myosin, their thin and thick filaments, are not highly organized. So let's start off by focusing on skeletal muscle. So this is just a little piece of a skeletal muscle fiber. Here's one nucleus out near the sarcolemma. And there are a couple of other structures we need to be aware of. So we have these yellow structures. These are your T-tubules. Now, the function of T-tubules is to bring the action potential that travels along the sarcolemma down into the depths of your muscle fiber. So essentially, it is a connection of the sarcolemma, but it kind of digs into the middle of your muscle fiber. Then we have these big blue structures. These are your sarcoplasmic reticulum, and they are filled with an important ion. So this ion is going to be the most important ion for all muscle contractions, and it is calcium. So calcium is going to be the big player. And then notice there's a lot of mitochondria so that we have enough energy to contract. And then if we take a look at our myofibril, we can see our thin and thick filaments. So these are your actual contractile proteins. So let's take a closer look. So they've taken your sarcomere and we've just named the different parts essentially. Okay, so here we have our Z disc. So Z disc to Z disc is one sarcomere. And your thick filament is the purple in the middle. Your thin filament is the pink in the edges. And so if we blow up our sarcomere, this is what it looks like. Okay, so again, here's our Z disc to Z disc. Our thick filament in purple, our thin filament in pink. The part of the entire thick filament, so everything in purple, is known as your A band, and we can see the A band here. And so when your muscle contracts, the A band is the only thing that does not change length. It's always going to stay the same, and that's because your thin and thick filaments don't actually coil up like a spring when you contract. They stay the same length, they just increase overlap. We have then the H zone. The H zone is just the thick filament that's not overlapping with the thin. And then the I band is just the thin filament that's not overlapping with the thick. So if we take a closer look at our thick filament, one thick filament is actually made up of 250 individual myosin molecules. And a myosin molecule has a tail, a hinge, and a head. So the hinge does what the name says. It hinges. And the head, the myosin head, is really important. This is where ATP is going to bind, and this myosin head is what's going to bind to our thin filament. So our thin filament over here is made up of several proteins. We have these round globe looking things. These are your G-actin molecules, and we kind of have two strings of them kind of coiled around each other. And then we have this long filament here that kind of travels all along our G-actin molecules, and that's known as our Tropomyosin. And then we have these little proteins that are called our troponin, and they're all going to be very important in muscle function. So other structures that we find in our sarcomere, we have our titin and nebulin. So nebulin is shown here in blue. It helps align your thin filament, your actin. So your actin has something that it's kind of attached to. And then your thick filament has titin. So that's the green. And the titin also provides elasticity as well as obviously stabilizing the myosin. So for a muscle to contract, several things have to happen. There has to be events at the neuromuscular junction. So neuromuscular junction is the junction or the connection between the somatic motor neuron and the muscle. And what happens here, we're going to have excitation contraction coupling, meaning how do we get from exciting our muscle with an action potential, with a stimuli from our somatic motor neuron, to actually getting a contraction? So how does that coupling occur? And we'll go over that. I said calcium is going to be a big player. So calcium is going to be a signal for the contraction to occur. And then you'll have your contraction relaxation cycle. A couple of terms just to be aware of. We have our muscle tension. That's the force that's created by the muscle. A load is the weight that opposes contraction. Contraction is when the muscle is generating tension or generating force. And then relaxation is when the muscle is releasing tension or releasing force. So when a sarcomere contracts, I said this earlier, the A band is the only thing that stays constant. Everything else gets shorter. And basically when a sarcomere contracts, your thin and thick filament are increasing overlap. They are sliding past each other. This is known as the sliding filament theory. Okay, so we have our relaxed muscle sarcomere, and then here is what it looks like when it's contracted. Now, what's actually allowing the sarcomeres to slide past each other? So that's where we're going to get to here. So here is our myosin head. So this is just one of the 250. So the myosin head is where ATP binds, and the myosin head is who reaches up to grab our thin filament. Now, if we take a look at our thin filament, here's our G-actin molecule. Now, the myosin head wants to bind the G-actin molecule, but it only wants to bind it on a particular site. In this case, it's the site that's shown in yellow. Now, this site that myosin head wants to bind to is known as the myosin binding site on the G-actin molecule. It's literally what it is. It's the site that myosin wants to bind to on the G-actin molecule. However, it can't do that right now because this long filament here, this tropomyosin, is in the way. And then here's our little troponin. So let's see why that guy is important. When a muscle is stimulated to contract, calcium, remember that's the big player, from the sarcoplasmic reticulum gets released. When calcium gets released from the SR, the sarcoplasmic reticulum, calcium is going to bind to this little guy, troponin. And when calcium binds troponin, it's going to cause troponin to undergo a conformational change. Basically, it's going to cause troponin to change shape. And when it changes shape, because it is physically connected to tropomyosin, it also moves tropomyosin out of the way. And so now the myosin binding site on the G-actin molecule is exposed, so the myosin head will reach up and form a crossbridge. A crossbridge is just a connection between the thin and thick filament, and then you will have your power stroke. And that's when the hinge does its job, which is to hinge. And that's your contraction. So where does ATP come in? So we know that we need ATP for muscles to contract. Well, the binding of ATP to the myosin head is what actually causes the myosin head to release from actin. Okay, so the actual binding, when ATP binds to the myosin head, it weakens the connection, and so you get the release. And then what you're going to do is you're going to use the energy of ATP to recock the myosin head. So the myosin head, notice it was kind of pointed inward here. Now that you've used the energy of ATP, notice ATP is now ADP down here. Now the myosin head is recocked, and it's ready to bind again. And because there's no tropomyosin here, because the binding site is exposed, it will again form a crossbridge and undergo a power stroke. And again, you'll stay contracted or you'll stay bound until a new ATP comes to then cause release. Okay, so let's see how we go from actually exciting our skeletal muscle fiber to getting that contraction. So this is known as excitation contraction coupling. We're going to couple exciting our muscle to a contraction. So here is our somatic motor neuron. So our somatic motor neuron, the action potential traveled down the somatic motor neuron. This one is myelinated, by the way, with our Schwann cells. And once the action potential, which we know is just the movement of sodium and potassium, sodium in, potassium out, once it reaches the axon terminal, voltage-gated calcium channels open. Calcium will enter into the terminal and cause these vesicles to exocytose. Now, our somatic motor neurons release the neurotransmitter acetylcholine, and it's going to release it into the synaptic cleft. So this is our neuromuscular junction because it's the connection between the neuron and the muscle. Now, where the neuron innervates the muscle part is actually specialized. So notice where the neuron is innervating the muscle, we have these little folds, these invaginations, and that's to increase surface area. So this area of the neuromuscular junction, the muscle part of the neuromuscular junction, is called the motor end plate. So the acetylcholine gets released, and remember, acetylcholine binds to acetylcholine receptors, or we can call them cholinergic receptors, and there are different isoforms. We have our nicotinic and our muscarinic. And on our skeletal muscle, it's actually the nicotinic receptors. So acetylcholine will bind to these nicotinic receptors. Two of them actually have to bind to each receptor, and that causes the receptor to open. And when the receptor opens, sodium rushes in and potassium rushes out. Now, more sodium rushes in than potassium rushing out. So basically, we have pluses coming in and pluses going out, but more pluses are coming in than going out. And so our resting membrane potential, which is at -70, starts to depolarize. And if it depolarizes to threshold, then we're going to fire an action potential along our sarcolemma of our muscle. And so here's our action potential traveling along the sarcolemma. And then guess what? It comes across a T-tubule. Remember, a T-tubule is basically an invagination of our sarcolemma, and it brings the action potential from out here in the surface into the depths of our muscle fiber. Now, right up against our T-tubules are our sarcoplasmic reticuli. So there's going to be one on either side. So when the action potential travels down the T-tubule has an important receptor here called the DHP. DHP stands for dihydropyridine receptor, and it is a voltage-gated channel. So when the action potential travels right past this DHP receptor, and which we know an action potential is a huge change in voltage, this DHP receptor will change shape. Now this guy won't actually open, meaning nothing's going to travel through this guy, but he will change shape. Now take a look. This DHP receptor is physically connected to this receptor here on the sarcoplasmic reticulum called the RIR. And RIR stands for ryanodine receptor. This ryanodine receptor is mechanically gated. Remember, mechanically gated channels can get pulled open essentially. And that's what's going to happen. When the DHP receptor changes shape, because it is physically connected to the RIR, it pulls the RIR open. So then what happens? Well, the ryanodine receptor, when it gets pulled open, it allows all the calcium that's inside the sarcoplasmic reticulum to now diffuse out into the cytosol of our muscle. And so what happens to calcium? We already talked about that. Calcium will bind to troponin. Troponin will change shape, moving tropomyosin out of the way, and now our myosin head will form a crossbridge with our G-actin molecule, and then we'll have our power stroke, and that is our contraction. So this is how we went from exciting our muscle to getting our contraction. Now that we've contracted, we now need to relax. To relax, we have to put calcium back where it came from. It came from the sarcoplasmic reticulum, so we need to pump it back into the SR. The word pump means we need ATP to do this. Why? Because we're trying to pump a lot of calcium into the SR, which already has a high concentration of calcium, so we need energy to do that. We also need energy, don't forget, here in the myosin head. Remember, after a power stroke, the myosin and actin will stay attached until a new ATP comes to bind. When new ATP binds, it will release, and then you'll use the energy of ATP to recock the myosin head for the next crossbridge cycle. We also need ATP up here in the sodium-potassium pump. Remember, an action potential is where sodium rushes in, potassium rushes out. Well, we got to get those ions back to where they came from, and our sodium-potassium pump will do that. It'll pump 3 sodium out and 2 potassium in. So obviously we need a lot of ATP for muscles to contract. And there are different ways we can get that ATP. One way is through phosphocreatine. So when we're at rest, when we make ATP through metabolism, aerobic metabolism, we may not need all of that ATP. And so what we're going to do is we're going to take the energy in ATP and attach it to creatine. The enzyme creatine kinase will do that for us. So if we take one of the phosphates out of a TP, triphosphate, we end up with a DP, diphosphate. And all we did was we took one of the phosphates and attached it to creatine, so now we have phosphocreatine. Then when we start working our muscles, we start exercising. In this case, you went from lounging to mowing your lawn. Now we need energy. So then all we'll do is we'll do this reaction in the opposite direction. We'll take the phosphate off of phosphocreatine and attach it back onto ADP. Again, the enzyme creatine kinase can do that for us. So now we're left with creatine because we took the phosphate away, and now instead of ATP, we have ATP. So this is a really quick way for us to make energy. It provides enough ATP for about 15 seconds of activity, around there. And again, why do we need ATP? We need it for the myosin head. We need it for the calcium pump. We need it for the sodium-potassium pump. Now, eventually our muscles will fatigue where they can't contract anymore. And there are a lot of reasons for fatigue. Now, loss of ATP, depletion of ATP is obviously one because we just demonstrated how we need energy for muscles to contract. But notice there's a lot of other ways or reasons for fatigue. And that's because the process of moving requires a lot or involves a lot of steps. So our central nervous system makes the decision. It has to send that information down the spinal cord through the somatic motor neuron, and then we have to have things happening in the neuromuscular junction, like the release of acetylcholine, activation of receptors. Then we need the exciting of our muscle to eventually lead to a contraction. That includes changes in membrane potentials and obviously the release of calcium. So a lot of steps are involved. And so if anywhere in this process it stops working, then we have fatigue. So we can even have psychological fatigue where the brain says it's not going to send any more action potentials to move as a protective response. Or maybe we just run out of acetylcholine. There are no more vesicles ready to be released. Or maybe we ran out of calcium, or troponin, the calcium-troponin interaction has reduced. Or we've built up phosphocreatine or ATP. We've depleted phosphocreatine, ATP, and glycogen, so we don't have enough energy. Or we've built up hydrogen ions or inorganic phosphate or lactate that's preventing our muscles from working even further. So lots of reasons for fatigue, although loss of ATP is the one that we're most familiar with. Now, the purpose of muscle is to contract and generate force, and our skeletal muscles can generate different amounts of force depending on what our purpose is. If we need to pick up something heavy, we need more force. If we need to pick up something light, we need less. And so one way we can change the amount of force our muscles produce is through recruiting motor units. So this is known as motor unit recruitment. So the first thing is to define a motor unit. A motor unit is a motor neuron and all of the fibers that it innervates. So if we take a look here, we have a purple, a blue, and a red motor neuron. These are your somatic motor neurons. And notice the purple motor neuron innervates all of the purple fibers. The blue motor neuron innervates all of the blue muscle fibers, and the red motor neuron innervates all of the red fibers. That means whenever the purple motor neuron fires an action potential, it will always innervate all of the purple. So notice they are always working together just like a unit. So again, the purple motor neuron and all of the purple fibers that it innervates is a motor unit. When we need to generate more levels of force, we can stimulate multiple motor units at the same time. So we can recruit the purple and the blue to stimulate together. And when we do that, we're going to have all of the purple and all of the blue fibers contracting together. That's a lot of fibers. That's going to be more force. If that's not enough, we can even recruit the red motor neuron. Then we'll have all of these muscles contracting together. So by recruiting more motor units, essentially we have more muscle fibers participating in the contraction, and as a result, we'll be able to generate more force. Now, another thing that influences the amount of force a muscle produces is the length of your muscle when you initiate the contraction. And that's because of this length-tension relationship. So these are your sarcomeres at different lengths. This sarcomere here, labeled C, this is your sarcomere at its optimum length. And the reason it's optimum in this length is because we have a lot of overlap between our thin and thick filaments, but we still have room to slide. So remember, a contraction is a sliding filament theory where our thin and thick filaments slide across each other and we increase overlap. So at this length, sarcomeres are at the perfect length where we have overlap. So we're going to be able to have a lot of cross bridges, and we have room for the sarcomeres to actually slide past each other and contract, or the filaments to slide past each other and contract. From this optimum length, if we were to shorten our sarcomeres and then try to contract, we'll actually produce less force. And that's because in this shorter sarcomere length, even though we have a lot of overlap, so we're going to have a lot of cross bridges, the problem is we have no room to slide. And the shorter our sarcomeres become, the less room to slide. Notice it's very crowded here, so we will actually produce very little to no force. From optimum length, if we were to then lengthen our muscle even more, our sarcomeres get stretched apart even further. So we'll have a lot of room to slide, but the problem is not as many cross bridges are able to participate. And as a result, we have less and less force being generated. So our muscles have an optimum length at which we're going to be able to produce the most force. So the length of your muscle, the length of your sarcomere when you contract also influences the amount of force you can produce. So in skeletal muscle, we're going to operate in this optimum length. I just want to point out now that our cardiac muscles also have sarcomeres, so this also applies to our cardiac muscles. And our cardiac muscles actually operate at a more shortened length, and we'll talk about why that is when we get to the heart. Now, another way we can change the amount of force our muscles produce is through summation. So here, every little triangle represents a stimuli, so an action potential. So we've stimulated our muscle and our muscles contracted and relaxed. We've waited and then stimulated again. We contract and relax. Now, our stimuli is over, so technically we could give another stimuli right here, and that's what we did. So we stimulated, we let the muscle contract, and before it's completely relaxed, we stimulated the muscle again. And what happens is the next contraction adds on top of the first. And we can keep doing that, and notice our contractions just keep adding on top of each other, and this is another way we can increase the amount of tension. Now, when we keep doing this, eventually we get to a point where we reach our maximum tension. That's because our muscles, at some point, no matter how much you stimulate it, that's the maximum force it can produce. Now, this type of tetanus here is known as an unfused tetanus, versus over here we have a fused tetanus. So the word fused means stuck together. So here we are stimulating our muscles. The frequency at which we are applying the contractions or the stimuli are so quick that the contractions are essentially sticking together. And so we are able to produce a nice smooth contraction. Over here, notice the stimuli, they're a little bit further apart, and so our muscles relax a little bit in between, and that's why we have an unfused tetanus here. Now, let's take a look at the different types of contractions we can have in our or we can produce with our skeletal muscle. We have isotonic and isometric. Iso means same. So in this case, tonic or tone is the same and the length can change. So an isotonic contraction is where you're going to change your muscle length. And when we change length, we can change it two ways. We can get shorter or longer. So when your muscles get shorter during a contraction, that's called a concentric or a shortening contraction. If it gets longer, it's called an eccentric or a lengthening contraction. So think of doing bicep curls. When you flex your arm and bring the weight up to your body, that's a concentric contraction. Your biceps are getting shorter. When you put down the weight and extend your arm, you're still contracting your biceps, but your biceps are getting longer. Then we have isometric contraction. Iso, in this case, metric, think of meters or length. So the length is going to stay the same. The force can change. So when you're holding something steady, or if you're pushing against something and that thing that you're pushing against doesn't move, then your muscles are contracting, but they're not changing length. So that's an isometric. Okay, so that's it for skeletal muscle. And here we just have what a skeletal muscle contraction looks like compared to cardiac and smooth. So if the red is our skeletal muscle contraction, notice cardiac muscle contractions last a little bit longer. They're a little bit slower, last a little bit longer. And then take a look at smooth. Smooth muscle contractions are a lot slower and they last a lot longer. And so that's what we're going to move on to next, smooth muscles. So because smooth muscle contractions are so long, they use less energy. And as a result, we have less oxygen consumption for smooth muscle contractions because, again, they can maintain their force for long periods of time. So the ATPase activity is much slower here. There's also something called myosin light chain. In skeletal muscle, we have it there too, but they're not that important there, so we don't really discuss it. But in smooth muscle, this myosin light chain plays a really important regulatory role. So this is going to be a really important part of smooth muscle contraction. Actin is a lot more plentiful. There's less sarcoplasmic reticulum because in smooth muscle, we also get calcium from the extracellular fluid. There are no sarcomeres here. That's why it's smooth looking. There's no distinct organization. And smooth muscle cells can be activated many ways. So skeletal muscle is only activated by somatic motor neurons, but smooth muscles can be activated by stretch receptors. They can they can be activated by hormones, by autocrines, paracrines, and they can activate all by themselves, and that's known as myogenic contraction, where they contract kind of all by themselves. So smooth muscle isn't studied as much, and there are some reasons for that. First is there's a lot of variety. For example, we have single unit versus multi-unit structure. So if you look here in the small intestine, all of these smooth muscle fibers are connected to each other through gap junctions. And as a result, they act like a single unit, right? So if you activate one or two, those depolarizations can spread from one to the other. So they can all work together like a single unit. Over here in the eye, there are no gap junctions, so these are known as multi-unit. So if you want to activate these muscles, you have to activate them individually. Also, anatomically, it makes it a little bit more difficult to study because they kind of contract in this big old blob. So if you take a look, unlike our skeletal muscle, which is kind of long, thin, rod looking, and so they contract kind of, the contraction is pretty neat. Notice it's not the case here. So it's a little bit more difficult to study. And it's regulated, like I said, many different ways. So smooth muscles can be innervated by the sympathetic nervous system. The parasympathetic can tell it to contract. Hormones, paracrines. Paracrines are just chemicals that are released by one cell that acts on a neighboring cell. It could be activated by autocrines. That means the smooth muscle will actually release a chemical that acts on itself to tell it to contract. And then again, it can undergo myogenic contractions where they contract all by themselves. So let's see how we go from exciting our smooth muscle to contracting. So this is our second excitation contraction coupling slide. Previously it was on skeletal, now we're looking at smooth. And notice, just looking at it, it looks very different. Now, however you activate smooth muscle, whether it's through hormones or the sympathetic or the parasympathetic, it all starts with extracellular calcium coming in to the smooth muscle. So calcium from the extracellular fluid will come into the smooth muscle, and that will trigger more calcium to be released from the sarcoplasmic reticulum. So now we're going to have this increase in calcium from both the SR and the ECF, and now calcium is going to bind CAM. This stands for calmodulin. So here is calcium bound to calmodulin. And when calcium and calmodulin are bound together, it will activate MLCK. So MLCK stands for myosin light chain kinase. And myosin light chain kinase is normally inactive. But when calcium and calmodulin are bound together, it will activate myosin light chain kinase. And what MLCK does is it puts a phosphate onto myosin light chain. So here's our myosin that you're familiar with. This is also known as myosin heavy chain. Right next to it, it's not shown here, is a little protein called myosin light chain. What this MLCK does, myosin light chain kinase, this kinase puts a phosphate onto the myosin light chain. So again, it looks like it's putting it onto the purple myosin, which is our myosin heavy chain, but it's actually putting it onto the little light chain that's next to it or attached to it. And when the myosin light chain has a phosphate attached to it, then this myosin head can form a crossbridge with actin. So there's no tropomyosin and troponin and all of that. Instead, what regulates whether or not the myosin head binds to actin is whether or not the little myosin light chain has a phosphate. So that's why that light chain plays an important regulatory role. So you're going to contract, and then to relax, you put calcium back where it came from. So calcium came from the sarcoplasmic, so you're going to pump it in there. It came from the ECF, so you'll pump it out there. As you lose calcium by pumping it into either the SR or into the ECF, you're going to lose it from the cytosol of your muscle, then calcium and calmodulin will start to unbind. And when they unbind, you will no longer have active myosin light chain kinase. Instead, the myosin phosphatase will start to then remove the phosphate off of the light chain. And now that the light chain doesn't have a phosphate, the myosin head and actin can't interact, and so you will relax. So that is smooth muscle. So very different than skeletal muscle. And that is it for this chapter. Thank you.